How do cells become cancerous?
Cancer is caused by the unrestrained growth of cells. Cells that do not follow the rules of normal cell cycling may eventually become cancerous. This means that the cells reproduce more often than normal, creating tumors. Usually this happens over an extended period of time and begins with changes at the molecular level. There are more than 100 distinct types of cancer, each of which behaves in a specific fashion and responds to treatment differently.
Which bryophytes are most closely related to green algae?
Hornworts are more closely related to green algae than to any other group of plants. Hornwort cells usually have a single, large chloroplast with a pyrenoid. Granular, starch-containing body, similar to those of green algae. Mosses and liverworts are like all other plants because they have many dish-shaped chloroplasts per cell.
Who made the first gene map?
Although gene maps are a relatively recent, past 30 years, means of locating genes. Geneticists of the early 20th century had prototype gene maps. Edmund Beecher Wilson, 1856 to 1939, and his colleagues were the first to demonstrate that the genetic differences between males and females were due to a special pair of chromosomes in the cell. Thomas Hunt Morgan, 1866 to 1945, Calvin Bridges, 1889 to 1938. And their colleagues were able to place a gene known to be inherited differently by males and females onto one of the sex chromosomes. This was the beginning of the first gene map.
Who first developed the modern technique for the isolation of cell parts?
Robert R. Bensley, 1867 to 1956, and Norman Lewis Hoare, 1902 to 1958, disrupted the liver cells in a guinea pig and isolated mitochondria in 1934. Between 1938 and 1946, Albert Claude, 1899 to 1983, continued the work of Bensley and Hoare and isolated two fractions, a heavier fraction consisting of mitochondria and another fraction of lighter submicroscopic granules, which he called microsomes. Further developments led to the development of centrifugal techniques of cell fractionation commonly used now. The development of this procedure was one of the earliest examples of differential centrifugation. It initiated the era of modern experimental cell biology.
What is the significance of Darwin's study of finches?
In his studies on the Galapagos Islands, Darwin observed patterns in animals and plants that suggested to him that species changed over time to produce new species. Darwin collected several species of finches. The species were all similar, but each had developed beaks and bills specialized to catch food in a different way. Some species had heavy bills for cracking open tough seeds. Others had slender bills for catching insects. One species used twigs to probe for insects in tree cavities. All the species resembled one species of South American finch. In fact, all the plants and animals of the Galapagos Islands were similar to those of the nearby coast of South America. Darwin felt that the simplest explanation for this similarity was that a few species of plants and animals from South America must have migrated to the Galapagos Islands. These few plants and animals then changed as they adapted to the environment on their new home, eventually giving rise to many new species. Evolutionary theory proposes that species change over time in response to environmental challenges.
What is divergent evolution?
When two species move away from the traits that they share with a common ancestor, as they adapt to their own environments, the result is called divergent evolution. As an example, imagine the diversity among bird species. Ducks, hummingbirds, ostriches, and penguins are all descended from an ancestral bird species. Yet they have all diverged as they adapted to their particular environments.
How is the term mole used in chemistry?
A mole, is a fundamental unit of measure. It refers to either the gram atomic weight or the gram molecular weight of a substance. A mole is equal to the quantity of a substance that contains 6.02 x 1023 atoms. Molecules, or formula units. This number is also called Avogadro's number, named after Amedeo Avogadro. 1776 to 1856, who is considered to be one of the founders of physical science.
Why do animals migrate?
Animals migrate for a variety of reasons, including climate. Too hot or too cold during part of the year, food availability, seasonal, and breeding. Some animals need a specific environment to lay eggs or give birth. Animals move between locations to take advantage of optimal environments.
What is the cytoskeleton and what is its function?
The cytoskeleton is a structural feature of eukaryotic cells that was revealed by advanced microscopy. It consists of an extensive three-dimensional network of interconnected filaments and tubules that extends throughout the cytosol, from the nucleus to the inner surface of the cell membrane. These filaments and tubules determine cell shape and facilitate a variety of cell movements.
If an allele is lethal, then why would it not disappear from a population totally?
In the example of cystic fibrosis, heterozygotes may be more resistant to life-threatening dehydration caused by diseases such as cholera, which is accompanied by massive diarrhea. In cholera, caused by the lethal bacterium Vibrio cholera, the bacteria produce a toxin that causes considerable loss of water, dehydration, and death within a short time. Another example of heterozygote advantage is the high frequency of heterozygotes for sickle cell anemia in Africa, and increased resistance to malaria.
Which animal has the longest gestation period?
The animal with the longest gestation period is not a mammal, it is the viviparous amphibian. The alpine black salamander, which can have a gestation period of up to 38 months at altitudes above 4,600 feet. 1,402 m, in the Swiss Alps, it bears 2 fully metamorphosed young.
What is balanced polymorphism?
When a trait exists in several forms within a population, it is said to be polymorphic. Polymorphisms that maintain a stable distribution within the population over generations are known as balanced polymorphisms. Balanced polymorphism can be maintained if heterozygotes, mixtures of two types, have a fitness advantage. When this occurs, both types of alleles are maintained in the population. A classic example of this is sickle cell anemia. Individuals who are heterozygous, HH, are resistant to malaria, dominant homozygotes. HH, are susceptible to malaria, and recessive homozygotes, HH, have sickle cell anemia. Because those who have both types of alleles and who live in malaria-prone regions are the most likely to survive long enough to produce children. Both types are maintained in the population at a relatively stable rate.
How do statin drugs affect cell function?
Statins are a group of drugs that work to lower cholesterol levels, particularly the bad cholesterol, low-density lipoprotein known as LDL. The drugs work in two ways. 1. They block an enzyme that is needed for cholesterol production. 2. They increase LDL membrane receptors in the liver. Cholesterol can only get into cells by binding to specific receptors that remove the LDL from blood. The extra receptors that statins create help decrease the cholesterol levels. As Americans become more aware that high cholesterol is a major risk factor for heart disease, statins are becoming increasingly popular.
Is there an average lifespan of a cell?
No. Most living cells do not live longer than a month. Even cells that live longer, such as liver and brain cells, constantly renew their components so that no part of the cell is really more than a month old. One example of a cell with a longer lifespan is the memory B cell. Memory B cells can survive for several decades, even a lifetime, providing an immune response to pathogens invading for a second time.
How many species of fungi are plant pathogens?
More than 8,000 species of fungi cause disease in plants. Most diseases found among both cultivated and wild plants are caused by fungi. Some pathogenic fungi grow and multiply in their host plants. Others grow and multiply on dead organic matter and host plants. Fungi that are pathogenic to plants can occur below the soil surface, at the soil surface, and throughout the body of a plant. Fungi are responsible for leaf spots, blights, rusts, smuts, mildews, cankers, scabs, fruit rots, galls, wilts, tree diebacks and declines, as well as root, stem, and seed rots.
How does ozone benefit life on Earth?
Ozone, a form of oxygen with 3 atoms instead of the normal 2, is highly toxic. Less than 1 part per million of this blue-tinged gas is poisonous to humans. In Earth's upper atmosphere, stratosphere, ozone is a major factor in making life on Earth possible. About 90% of the planet's ozone is in the ozone layer. The ozone belt shields and filters Earth from excessive ultraviolet, UV, radiation generated by the sun. Scientists predict that a diminished or depleted ozone layer could lead to increased health problems for humans, such as skin cancer, cataracts, and weakened immune systems. Increased UV can also lead to the reduced crop yield and disruption of aquatic ecosystems, including the marine food chain. While beneficial in the stratosphere, near ground level ozone is a pollutant that helps form photochemical smog and acid rain.
What is ultrasound?
Ultrasound is another type of 3D computerized imaging. Using brief pulses of ultra-high frequency acoustic waves, lasting 0.01 second, it can produce a sonar map of the imaged object. The technique is similar to the echolocation used by bats, whales, and dolphins.
What wildflower was used by Native Americans to make red dye?
Native Americans painted their faces and dyed their clothes red with the root of the bloodroot wildflower. Sanguinaria canadensis, which is also called redroot, Indian paint, and tetawort. Bloodroot, found in shady, damp, and woodsy soils. Blooms in May and has white flowers that are 2 in 5 centimeters wide.
How is chromatography used to identify individual compounds?
Chromatography is another technique used to separate mixtures into their individual components. All methods of chromatography share common characteristics. The process is based on the principle that different chemical compounds will stick to a solid surface or dissolve in a film of liquid, to different degrees. Chromatography involves a sample, or sample extract, being dissolved in a mobile phase, which may be a gas, a liquid, or a supercritical fluid. The mobile phase is then forced through an immobile, immiscible stationary phase. The phases are chosen such that components of the sample have differing solubilities in each phase. The least soluble component is separated first, and as the separation process continues, the components are separated by increasing solubility.
What is cell fluorescence?
Fluorescence is luminescence caused by a pigment molecule. Pigments are molecules that absorb some colors of light while reflecting others. For example, green pigments, like those in leaves, absorb red and blue wavelengths of light but reflect green wavelengths. After being energized by photons, units of light energy, the electrons of some pigment molecules actually give off light as they fall back to their normal state. Chlorophyll molecules that play a role in photosynthesis have this ability, as do molecules found in several organisms, such as jellyfish. The body of the insect Photinus pyrolis, better known as a lightning bug, has the enzyme luciferase, which generates the chemical reaction that leads to a drop in the energy state of electrons A. Reaction similar to that which occurs in a chlorophyll molecule, allowing the insect to glow.
What is a flavor savr tomato?
The flavor savr tomato was produced in response to consumer complaints that tomatoes were either too rotten to eat when they arrived at the store or too green. Growers had found that they could treat green tomatoes in the warehouse with ethylene, a gas that causes the tomato skin to turn red. However, the tomato itself stayed hard. In the late 1980s, researchers at Colgene, a small biotech company, discovered that the enzyme polygalactouronase, PG, controlled rotting in tomatoes. The scientists reversed the DNA sequence of PG. The effect was that tomatoes turned red on the vine, and yet the skin of the tomatoes remained tough enough to withstand the mechanical pickers. However, before the Flavre Savre tomato was introduced to the market, Colgene disclosed to the public how the tomato was bioengineered. This caused a public protest that led to a worldwide movement against genetically modified organisms, GMO.
What does the term hydroponics mean?
This term refers to growing plants in some medium other than soil. The inorganic plant nutrients, such as potassium, sulfur, magnesium, and nitrogen, are continuously supplied to the plants in solution. Hydroponics is mostly used in areas where there is little soil or unsuitable soil, since it allows precise control of nutrient levels and oxygenation of the roots. It is often used to grow plants used for research purposes. Julius von Sachs, 1832–1897, a researcher in plant nutrition, pioneered modern hydroponics. Research plants have been grown in solution culture since the mid-1800s. William Garrick, a scientist at the University of California, defined the word hydroponics in 1937. In the 50 years that hydroponics has been used on a commercial basis, it has been adapted to many situations. NASA will be using hydroponics in the space station for crop production and to recycle carbon dioxide into oxygen. Although successful for research, hydroponics has many limitations and may prove frustrating for the amateur gardener.
How do aseptic procedures prevent contamination?
The goal of aseptic procedures is to keep the organisms used in an experiment separate from the millions of other organisms in the environment. These procedures focus on the way to transfer organisms from test tube to test tube, from test tube to flask or petri dish, and from petri dish to petri dish or flask.
Why does the size of the nucleolus vary in cells?
Cells with a high rate of protein synthesis have a large number of ribosomes. In these active cells, nucleoli tend to be large and can account for 20 to 25% of the nuclear volume.
How are truffles used in cooking?
Dark truffles are generally used to flavor foods such as omelets, polentas, risottos, and sauces. White truffles are usually served raw, they are often grated over foods such as pasta or dishes containing cheese, as their flavors are complementary. They are also added at the last minute to cook dishes.
What is DNA acetylation?
DNA acetylation refers to the addition of an acetyl, CH3 group to one of the histone proteins that help hold DNA in its tightly wound configuration. When hist1s are altered by this change, the binding between hist1s and DNA is relaxed. This promotes transcription in eukaryotic cells.
How long do animals, in particular mammals, live?
Of the mammals, humans and fin whales live the longest. Below is the maximum lifespan for various animal species.
How are seedless grapes grown?
Since seedless grapes cannot reproduce in the manner that grapes usually do, i.e., dropping seeds, growers have to take cuttings from the plants, root them, and then plant the plant cuttings. Seedless grapes come from a naturally occurring mutation in which the hard seed casing fails to develop. Although the exact origin of seedless grapes is unknown, they might have been first cultivated thousands of years ago in present-day Iran or Afghanistan. Currently, 90% of all raisins are made from Thompson seedless grapes.
What are chiasmata?
The site of crossing over, where homologous chromosomes actually exchange material during the first round of meiosis, is known as a chiasma. The plural version is chiasmata.
Can we make artificial cells?
Research in progress at the National Aeronautical and Space Administration, NASA, is focused on artificial cells as a means to deliver medicine in outer space. These cells are able to withstand dehydration and thus can be safely stored for long periods. Artificial cells are made of a polymer that acts like a cell membrane. But the polymer is stronger and more manageable than real membranes. These polymers are called polymersomes and can be made to cross-link with other polymers. Researchers feel that many different kinds of molecules can be encapsulated within these polymersomes and then delivered to specific target organs. An example would be an artificial blood cell that not only delivers oxygen but also medication as it travels through the body.
What products does 1 acre of trees yield when cut and processed?
There are approximately 660 trees on 1 acre of forest. This number of trees can yield approximately 105,000 feet. 32,004 m of lumber, more than 30 tons, 30,000 kilograms, of paper, or 16 cords of firewood.
What is biogeography?
Biogeography is the study of the distribution, both current and past, of individual species in specific environments. One of the first biogeographers was Carolus Linnaeus, 1707 to 1778. A Swedish botanist who studied the distribution of plants. Biogeography specifically addresses the questions of evolution, extinction, and dispersal of organisms in specific ecosystems.
Is alcoholism an inherited disease?
If all of the adults in the U.S. were to be tested, an estimated 10% of the adult population would be classified as alcoholic. Of that number, the ratio of males to females is approximately 4 to 1. This disproportion could be attributed to both environment and genetics. Research involving male relatives, both biological and adoptive, points to a multifactorial source for alcoholism. In other words, while sons and brothers of male alcoholics are likely to be alcoholic as well, 25% and 50%, respectively. Genetic inheritance is not solely responsible for all of the cases of alcoholism. Alcoholism has been linked to depression, as more is learned about the genetic basis of mood disorders. The genetic basis of alcoholism may be unraveled.
What is the Benedict's test for reducing sugars?
A commonly used test to detect carbohydrates is the Benedict's test for reducing sugars such as glucose and fructose. Benedict's reagent, containing sodium bicarbonate, sodium citrate, and copper sulfate, is added to a solution and heated. The Benedict's test identifies reducing sugars based on their ability to reduce the cupric ions to cuprous oxide at high pH values, basic solutions. Cuprous oxide is green to reddish-orange. A green solution indicates a small amount of reducing sugars, while a reddish-orange solution indicates an abundance of reducing sugars. If the solution contains sucrose, a non-reducing sugar, there is no change in color in the solution, and it remains blue.
Which organism is known as the humongous fungus?
The humongous fungus is an enormous, underground fungus that grows in northern Michigan. The fungus, Armillaria gallica, was discovered in 1992. It encompasses 35 acres, 15 hectares, and is thought to be at least 1,500 years old. Scientists proved that the organism was a single fungus by taking 20 samples of the fungus and performing DNA analyses on 16 fragments from each sample. The genetic material of each sample was identical.
Can animals recognize different languages?
Scientists have compared language discrimination in human newborns and cotton-top tamarin monkeys. Each group was presented with 20 sentences in Japanese and 20 sentences in Dutch. Infant reactions were gauged by their interest in sucking on a pacifier. When infants first heard sentences in Dutch, they sucked rapidly on their pacifiers. But after a while they grew bored with the Dutch sentences, and the rate of sucking slowed. When someone started speaking in Japanese, they showed increased interest by increasing their rate of sucking. Language discrimination was studied in tamarin monkeys by changes in the facial orientation toward or away from the loudspeaker, similar to the infant reactions. The monkeys looked at a loudspeaker broadcasting Dutch sentences and looked away when bored. When someone started speaking Japanese sentences, they looked back at the loudspeaker. Results indicate shared sensitivities between monkeys and humans in the ability to discriminate between languages.
Why do cows have 4 stomachs?
Cows have 4 stomachs in order to process their low-quality diet of grass. The 4 sections are, 1. The rumen, 2. The reticulum, 3. The omasum, and 4. The abomasum. Cows are a type of mammal known as a ruminant. Ruminants eat rapidly and do not chew much of their food completely before they swallow it. The liquid part of their food enters the reticulum first, while the solid part of their food enters the rumen, where it softens. Bacteria in the rumen initially break the food material down as a first step in digestion. Ruminants later regurgitate the partially liquefied plant parts into the mouth, where they continue to munch it in a process known as chewing their cud. Cows chew their cud about 6 to 8 times per day, spending a total of 5 to 7 hours in rumination. The chewed cud goes directly into the other chambers of the stomach, where various microorganisms assist in further digestion. Herbivores have a longer small intestine to allow maximum time for the absorption of nutrients.
What factors have contributed to the success of seed plants?
Seed plants do not require water for sperm to swim to an egg during reproduction. Pollen and seeds have allowed them to grow in almost all terrestrial habitats. The sperm of seed plants is carried to eggs in pollen grains by the wind or animal pollinators such as insects. Seeds are fertilized eggs that are protected by a seed coat until conditions are proper for germination and growth. Seeds are also dispersed by wind or animals.
What is meant by a diploid organism?
A diploid organism is one that has two sets of chromosomes, one from each parent.
What are the basic steps to transfer bacteria using aseptic procedures?
First, plug the test tube or flask with cotton or plastic cap. Sterilize the inoculating loop or needle, tool used to transfer the bacteria, by heating the wire in the flame of a Bunsen burner or alcohol lamp until red hot. Remove the cotton plugs from the test tubes or flasks, and briefly flame the mouths of the test tubes or flasks. Pick up the organisms to be transferred by dipping the inoculating loop or needle into the liquid culture or touching the growth lightly. Transfer the culture to a new test tube by dipping the loop gently into the broth or drawing it across the agar. Pass the mouths of the tubes through the flame again and then reinsert the cotton plugs to prevent the entrance of other microbes. Heat the inoculating loop or needle again to destroy any remaining organisms. The procedure to transfer a sample to a petri dish is similar except the culture is streaked along the agar in a pattern to isolate the bacteria on the petri dish.
According to the most recent estimate, how many genes do humans have?
As of April 2003, researchers estimated that building a human would require only about 24,500 genes. The mustard seed plant has about 25,000 and fruit flies about 13,000. When analysis of the draft human genome sequence was published in February 2001, there was estimated to be only about 30,000 to 40,000 protein-coding genes. This number will probably fluctuate as applications of the HGP are implemented.
What is the hydrologic cycle?
The hydrologic cycle takes place in the hydrosphere, which is the region containing all the water in the atmosphere and the Earth's surface. It involves 5 phases: condensation, infiltration, runoff, evaporation, and precipitation.
What is sexual selection?
when individuals of the same sex differ in their mating success and that difference is correlated to the presence or absence of a particular trait, that trait is said to be the result of sexual selection. Traits that provide no benefit to survival but that increase the likelihood of the male acquiring a mate are examples of sexual selection.
Who developed plant breeding into a modern science?
Luther Burbank, 1849 to 1926, developed plant breeding as a modern science. His breeding techniques included crosses of plant strains native to North America and foreign strains. He obtained seedlings that were then grafted onto fully developed plants for an appraisal of hybrid characteristics. His keen sense of observation allowed him to recognize desirable characteristics, enabling him to select only varieties that would be useful. One of his earliest hybridization successes was the Burbank potato, from which more than 800 new strains and varieties of plants including 113 varieties of plums and prunes were developed. More than 20 of these plums and prunes are still commercially important today.
Which aquatic animals have internal fertilization?
Certain sharks, skates, and rays have internal fertilization. The pelvic fins are specialized to pass sperm to the female. In most of these species the embryos develop internally and are born alive.
What is pasteurization?
Pasteurization is the process of heating liquids, such as milk, to destroy microorganisms that can cause spoilage and disease. This process was developed by Louis Pasteur, 1822 to 1895. As a method to control the microbial contamination of wine. Pasteurization is commonly used to kill pathogenic bacteria, such as Mycobacterium, Brucella, Salmonella, and Streptococcus, common to milk and other beverages. There are 3 methods for pasteurizing milk. In the first method, low temperature holding, LTH, milk is heated to 145 degrees Fahrenheit 62.8 degrees Celsius for 30 minutes. In the second method, high temperature short time, HTST, milk is exposed to a temperature of 161 degrees Fahrenheit 71.7 degrees Celsius for 15 seconds. This technique is also known as flash pasteurization. The most recent method allows milk to be treated at 286 degrees Fahrenheit 141 degrees Celsius for 2 seconds. This approach is referred to as ultra-high temperature, UHT, processing. Shorter-term processing results in improved flavor and extended product shelf life.
Which children's author studied and drew illustrations of fungi?
Beatrix Potter, 1866 to 1943, perhaps best known for having written The Tale of Peter Rabbit. In 1902, began drawing and painting fungi in 1888. She eventually completed a collection of almost 300 detailed watercolors, which are now in the Armitt Library in Ambleside, England. In 1897 she prepared a scientific paper on the germination of Agaricinae spores for a meeting of the Linnean Society of London. Although her findings were originally rejected, experts now consider her ideas correct.
Why are species of the genus Neurospora important?
Pink bread molds of the genus Neurospora have long served as powerful laboratory models used to study genetics, biochemistry, and molecular biology. Scientists first demonstrated the concept that one gene produces a corresponding protein by studying Neurospora. Its ease of growth and the extensive genetic information available for this organism make it a convenient model for the study of many processes found in higher plants and animals. Among the fungi, it is second only to yeast as a basic model organism.
What is the difference between a virus and a viroid?
Viroids are small fragments of nucleic acid, RNA, without a protein code. They are usually associated with plant diseases and are several thousand times smaller than a virus.
How many kinds of mushrooms are edible?
Among the basidiomycetes, there are approximately 200 varieties of edible mushrooms and about 70 species of poisonous ones. Some edible mushrooms are cultivated commercially, more than 844 million pounds. 382,832 metric tons, are produced in the United States each year.
What is a species?
There are several ways of defining a species, and scientists will use different definitions, depending on whether they are referring to a fossil, extinct, species or a living, extant, one. For example, an extant species can be defined as all the individuals of all the populations capable of interbreeding. A group of populations that are evolutionarily distinct from all other populations may also be defined as a species, even if they are incapable of interbreeding due to extinction.
What are homologous chromosomes?
In diploid organisms, chromosomes occur in matching pairs, with the exception of the sex chromosomes. Not all organisms are diploid, for example, bacteria have only one circular chromosome. And some insects may have an odd number of chromosomes. Every human somatic cell, excluding egg or sperm cells, has 46 chromosomes, 22 somatic chromosome pairs and one sex chromosome pair. The somatic chromosome pairs, called homologous chromosomes, carry the same sequence of genes for the same inherited characteristics.
Who introduced electrophoresis as an analytical technique?
Electrophoresis was introduced in 1937 by Arndt Salius, 1902 to 1971. A Swedish biochemist. His work pioneered the use of electrophoretic methods to separate chemically similar proteins of blood serum.
What is a microclimate?
When you notice that the temperature forecast in your local media is consistently warmer or colder than that which occurs in your neighborhood, you have identified a microclimate. Light, temperature, and moisture may all vary from one area to another within a biome because of changes in altitude, vegetation, or other factors.
What is the genetic code?
The genetic code is a chart depicting the relationship between each of the possible mRNA codons and their associated amino acids. The codons are grouped according to the amino acid they code for. Present in the code as well are the start and stop codons. The start codon actually codes for methionine, which is always the first amino acid in the polypeptide sequence. Methionine may appear elsewhere in the polypeptide as well. Methionine is removed during post-translational processing.
What does the term Umwelt refer to with regard to animal behavior?
Umwelt was first used by Jacob von Uexkell, 1864-1944, to describe the part of its environment that an animal can actually perceive with its sense organs and nervous system. Animals display this concept in the wild, but the same behavior can be seen with domestic animals. For example, if a stranger comes into your house and tries to pet your dog, this may be perceived as a threat within the dog's Umwelt. The practical side of this is that you should be aware of your pet's Umwelt when introducing it to new experiences and people.
How does carbon monoxide affect cell function?
Carbon monoxide is a highly poisonous gas. Because of its molecular similarity to oxygen, hemoglobin can bind to carbon monoxide instead of oxygen. And this subsequently disrupts hemoglobin's efficiency as an oxygen carrier. Carbon monoxide actually has a much greater affinity, about 300 times more, for hemoglobin than oxygen. When carbon monoxide replaces oxygen, this causes cell respiration to stop, leading to death. The particular danger of carbon monoxide poisoning lies in the fact that a person exposed to high levels of this toxin cannot be saved by being transporting to an environment free of the poison and rich with oxygen. Since the hemoglobin remains blocked, artificial respiration with overpressurized pure oxygen must first be performed to return the hemoglobin to its original function and the body to normal cell respiration.
Which organelle divides and compartmentalizes the cell?
The endomembrane system, which fills the cell and divides it into compartments, and is visible only through electron microscopy. The endoplasmic reticulum, ER, a series of interconnected membranous tubes and channels in the cytoplasm, is the largest and most extensive system of the internal membranes.
Since fungi lack the chlorophyll necessary to produce their own food, how do they obtain food?
Fungi are saprobes that absorb nutrients from waste and dead organisms. Instead of taking food inside its body and then digesting it, as an animal would, a fungus digests food outside its body by secreting strong, hydrolytic enzymes onto the food. In this way, complex organic compounds are broken down into simpler compounds that a fungus can absorb through the cell wall and cell membrane.
How does the root system of a monocot differ from the root system of a dicot?
The root system of a monocot is a fibrous mass of roots providing broad exposure to soil, water and minerals. The root system of a dicot consists of one large taproot, with many small secondary lateral roots growing out of it.
How do the excretory organs vary in different species?
Many animals, such as sponges, jellyfish, tapeworms, and other small organisms, do not have distinct excretory organs. Rather, they rid their bodies of waste through diffusion. Larger, more complex animals require specialized, often tubular, organs to rid their bodies of waste. For example, flatworms such as planarians have tubules that collect wastes and expel them to the outside via pores. Segmented worms such as earthworms have nephridia, tubules with a ciliated opening in each segment. Fluid from the body cavity is propelled through the nephridia. Wastes are expelled through a pore to the outside while certain substances are reabsorbed. Insects have a unique excretory system that consists of Malpighian tubules. Waste products enter the Malpighian tubes from the body cavity. Water and other useful substances are reabsorbed, while uric acid passes out of the body. Vertebrate animals have kidneys to dispose of metabolic wastes.
Is the same strain of yeast used to make lager and ale beers?
Two common strains of yeast are used to ferment beer: Saccharomyces carlsbergensis and Saccharomyces cerevisiae. Saccharomyces carlsbergensis, also known as bottom yeast, sinks to the bottom of the fermentation vat. Strains of bottom yeast ferment best at 42.8 to 53.6 degrees Fahrenheit, 6 to 12 degrees Celsius, and take 8 to 14 days to produce lager beer. Saccharomyces cerevisiae, also known as top fermenting yeast, is distributed throughout wort and is carried to the top of the fermenting vat by carbon dioxide, CO2. Top fermenting yeast ferments at a higher temperature, 57.2 to 73.4 degrees Fahrenheit, 14 to 23 degrees Celsius, over only 5 to 7 days. Top fermenting yeasts produce ales, porter, and stout beers.
Why was Wallace not as well known as Darwin?
While Darwin was well connected to the scientific establishment of the time, Wallace entered the scene somewhat later and so was less well known. Although Darwin would become far more famous than Wallace in subsequent decades, Wallace became quite well known during his own time as a naturalist, writer, and lecturer. He was also honored with numerous awards for his work.
What was Camillo Golgi's, 1843 to 1926, contribution to histology?
In 1873, Camillo Golgi, 1843 to 1926, devised a way to stain tissue samples with inorganic dye using silver salts. When he applied this technique and stained nerve tissue, he was able to see details previously not visible.
What is a peroxisome and what is its function in a cell?
Peroxisomes, also discovered by Christian de Duva, 1917, are surrounded by a single membrane and are the most common type of microbody in cells. They are especially prominent in algae, the photosynthetic cells of plants, and both mammalian kidney and liver cells. Peroxisomes contain detoxifying enzymes and produce catalase, which breaks down hydrogen peroxide into hydrogen and water.
What is the economic importance of roots?
Carrots, beets, turnips, radishes, horseradish, sugar beets, and sweet potatoes are all taproots that have been used as food for human consumption for centuries. The spices licorice, sassafras, and sarsaparilla. The flavoring used to make root beer, are derived from roots. The drugs aconite, gentian, ipecac, ginseng, reserpine, a tranquilizer, and protoveratrine, a heart relaxant, are all extracted from the roots of plants.
How does the iodine test detect starch?
Starch is a coiled polymer of glucose. Iodine reacts with the coiled molecules and turns bluish-black when added to a solution. A solution that remains a yellowish-brown color is a negative test for starch, whereas one that turns bluish-black is a positive test for starch.
What are the two types of euryhaline fishes?
There are two types of euryhaline, from the Greek terms euris, meaning broad, and hals, meaning salt, fishes. One type includes flounders, sculpins, and killifish that live in estuaries or intertidal areas where the salinity of the water fluctuates throughout the day. The second type includes salmon, shad, and eels that spend part of their life cycles in fresh water and the balance in seawater.
When offered a choice between plastic or paper bags for your groceries, which should you choose?
The answer is neither. Both are environmentally harmful, and the question of which is the more damaging has no clear-cut answer. On one hand, plastic bags degrade slowly in landfills and can harm wildlife if swallowed. And producing them pollutes the environment. On the other hand, producing the brown paper bags used in most supermarkets uses trees and pollutes the air and water. Overall, white or clear polyethylene bags require less energy for manufacture and cause less damage to the environment than do paper bags not made from recycled paper. Instead of having to choose between paper and plastic bags, you can bring your own reusable canvas or string containers to the store. And you can save and reuse any paper or plastic bags you get.
What is the Oparin-Haldane hypothesis?
In the 1920s, while working independently, Alexandre Oparin, 1894 to 1980, and John Haldane, 1892 to 1964, both proposed scenarios for the prebiotic conditions on Earth. the conditions that would have allowed organic life to evolve. Although they differed on details. Both models described an early Earth with an atmosphere containing ammonia and water vapor. Both also surmised that the assemblage of organic molecules began in the atmosphere and then moved into the seas. The steps of the Oparin-Haldane model are described below. 1. Organic molecules including amino acids and nucleotides are synthesized abiotically, without living cells. 2. Organic building blocks in the prebiotic soup are assembled into polymers of proteins and nucleic acids. 3. Biological polymers are assembled into a self-replicating organism that fed on the existing organic molecules.
What is the only four-horned animal in the world?
The four-horned antelope, Tetracerus quadricornis, is a native of central India. The males have two short horns, usually 4 in, 10 centimeters, in length, between their ears, and an even shorter pair, 1 to 2 in, 2.5 to 5 centimeters, long, between the brow ridges over their eyes. Not all males have 4 horns, and in some the second pair eventually falls off. The females have no horns at all.
What are the common types of slide preparations for investigation with a microscope?
Commonly prepared slide preparations are whole mounts, smears, squashes, and sections. Smears, squashes, and sections are techniques used to make specimens thinner or smaller. Whole mounts are often used to examine an entire organism or specific organ structure in some detail. Smears are mostly prepared for bacteriological and blood specimens. Squashes are prepared to study chromosomes. Sections are prepared to examine tissues and cells.
What is aneuploidy?
Cell division that results in an unequal separation of genetic material creates aneuploid. From the Greek terms an, meaning not, eu, meaning true, and ploid, meaning number, cells. Examples would be human cells with 47 or 45 chromosomes. For a diploid organism, aneuploidy is usually written algebraically as 2n plus 1 or 2n1, indicating an odd number of genes.
Is coral bleaching related to changes in the environment?
Although corals can capture prey, many tropical species are dependent on photosynthetic algae, zooxanthellae, for nutrition. These algae live within the cells that line the digestive cavity of the coral. The symbiotic relationship between coral and zooxanthellae is mutually beneficial. The algae provide the coral with oxygen and carbon and nitrogen compounds. The coral supplies the algae with ammonia, waste product, from which the algae make nitrogenous compounds for both partners. Coral bleaching is the stress-induced loss of colorful algae, zooxanthellae, that live in coral cells. In coral bleaching, the algae lose their pigmentation or are expelled from coral cells. Without the algae, coral become malnourished and die. The causes of coral bleaching are not completely understood, but it is believed that environmental factors are involved. Pollution, invasive bacteria such as Vibrio, salinity changes, temperature changes, and high concentrations of ultraviolet radiation, associated with the destruction of the ozone layer, all contribute to coral bleaching.
What are some commercial uses of plant hormones?
Plant hormones are used in a variety of applications to control some aspect of plant development. Auxins are used in commercial herbicides as weed killers. Another use of auxins is to stimulate root formation. It is often referred to as the rooting hormone and applied to cuttings prior to planting. Some hormones are used to increase fruit production and prevent pre-harvest fruit drop. Gibberellins are sprayed on Thompson seedless grapes during the flowering stage to thin the flowers on each cluster, thus allowing the remaining flowers to spread out and develop larger fruit. Gibberellins are also used to enhance germination and stimulate the early emergence of seedlings in grapes. Citrus fruits, apples, peaches, and cherries. When used on cucumber plants, gibberellins promote the formation of male flowers, which is useful in the production of hybrid seeds.